So I'm really excited to have you here today, Dr. Jiang. And I am Dr. Terry Jo Bichell nowadays. I'm a neuroscientist and the mom of a son who's now 22 years old with Angelman syndrome. And this is a joint production of the Foundation for Angelman Syndrome Therapeutics and the Angelman Syndrome Foundation to introduce families to scientists who are doing the cutting edge work for Angelman syndrome. So I'm very excited today to have you talk about your work that is funded by the FAST organization. So if you can introduce yourself, that would be great.
Sure. Thank you, Terry, to having me for this exciting sort of the joint venture together. So as you alluded already, my name is Yong-Hui Jiang. I'm here at Yale University School of Medicine. So I'm the professor in the genetics, neuroscience, and the pediatrics, and also the chief for the medical genetics. I may know many of you because I started working Angelman for quite a long time ago. Even when I was a student, we continue working on this very important topic, and I hope we can help the family like Terry, Jo, and many others.
So how long exactly have you been working on Angelman syndrome?
Oh yeah, very long. I started working at Angelman in 1995 when I was a student back to Baylor as Beaudet, Dr. Beaudet's PhD student. So I was studying working the Angelman project. So the major effort was to contribute to identify actually the Angelman UBC gene as my first effort. Then I created first Angelman mouse model that has been used by many over the last 25 years.
So were you around when the gene was discovered?
Oh yeah, I was a co-author for that gene was discovered.
That's pretty amazing. So you were with Dr. Beaudet at Baylor, Texas Children's, discovered the gene for Angelman syndrome, made the first mouse model. What are you doing right now?
So I moved to the year 2 years ago. So let me tell you a little bit of the history. So I finished all the training at Baylor and then moved to the Duke for 11 years. So I left the Duke end of 2019 and joined the Yale for the last 18 months. So the lab is still working on many different projects, but Angelman is still one of the major focus. So we study both continue Angelman mouse model, develop new treatment, testing new treatment with other company. For example, we're working with the company in Boston. It's a Sage Therapeutics, and they give us the novel compound we're testing in the animal model. The second project, which is a new project after I joined the Yale, working with FAST, which is created biorepository for the human patient-derived, Angelman patient-derived, we call it iPSC cell, which is induced pluripotent stem cell. Then eventually we're gonna also use the cell to create a called a brain organelle, which is called a mini brain. And then this is biorepository, eventually we'll be able to use it by all investigators, both in academic or commercial industrial laboratory, to testing new treatment for future.
So what you're doing right now for FAST, besides testing these compounds, is creating this biorepository. That's fantastic. So how does the biorepository work?
So yeah, so we are starting in the middle of last year during the COVID time. It definitely delayed a little bit, but we are making very good progress so far. I probably have contact or talking to many in the family listening to try to get your kids, Angelman kids, involved or participate in this project. Again, I've taken the opportunity to thank you all. Contact us, donated blood. So how it works, typically you contact us and we talking to you for the project. If you agree to, we actually develop modified protocols. We don't need you visit Yale due to the COVID. So we have you just collect the blood sample from with local pediatrician or any clinic, and then shipping to us. So once we receive the sample, we're working with Stem Cell Center here. It's one of the largest, first stem cell center at Yale in the world. Then we have a team here converting to the blood cell to the stem cell. Then we're testing them, make sure the quality is good. Then we then distribute anyone, any investigator interested in Angelman research. If a non-C cell, we freely given to them anywhere in the US or in the world.
Wow. So any person with Angelman syndrome can participate?
Yeah, so we try to making about the initial plan was fast support. We are planning to making 20 all different type, including the patient with a larger deletion, UPD, imprinting defect, UB3A mutation, both we call loss function or gain function. So we cover all different type of Angelman, which that's good, right? Every no family, Angelman family would be left out for this study. Of course, with the limited resource, we won't be able to do 100,000. So we only do 20. So then we're kind of screening for some criteria for the family. For example, we only picking the patient with older than 2 and younger than 18. Then we're also looking particular sort of a genetic report. We screen, so okay, yeah, you are eligible. Then we collect a blood. So that's everyone definitely can contact us, but we not guarantee everyone will eventually be on the candidate for the participation, but we're definitely happy to see you contact us. We screen for the eligibility.
And so once you get those 20 those 20 samples from 20 different patients that are, you know, depending on each type of genetic reason that the person has Angelman syndrome, then you're going to make those into stem cells?
Yeah.
And so
the first step, it goes to stem cell. And then the second step, actually more exciting, it goes to we call the brain organelle, and in the late term, basically making called mini brain, small brain, okay, which we truly allow myself, my team, to truly study the Angelman brain in the petri dish, which is a very, very new technique, only available for us to testing out over the last 5 years.
what does it look like in the dish? Does it look like a brain, or what does it look like exactly?
Under microscope, and by definition, that's all different brain structure you can see it at a cellular level. Of course, it's very tiny. It's like in the pea sort of the size of in the petri dish, but mimicking quite well for the brain structure, particularly for the cortical brain structure. That's the best one. But we are going to try to do a more sort of different brain region. Yeah, so it's amazing. That's where scientists get so excited. But you literally can study human brain in the petri dish, which is not possible, as I said, 5 years ago. But clearly, it's a new technique. It needs to be sort of getting better and better, and improve all different aspects. But clearly, this is the best if you want to study Angelman, because as you know, Angelman has this unique sort of brain-specific imprinting. That's no other replacement of anything. Even the mice, it's not going to be replaced the human brain research. That's where we get excited. Second is for any new kind of treatment, if you're testing effective in animal mice and other animals, so it's probably better to testing this system before you go to the FDA, because you are truly testing a human-derived neuron, and also we call the brain, not exactly the human brain, but it's the best that you can have. So if this is working, the chance to work in the human, in the real, it's much high compared. Oftentimes, we see many good things happen to the mice, and it just didn't translate well when it's moved to the human, because we know that limitation for the mice. Even as you know, I created the first model. I love working, continue working the mice, but there is a limitation. Mouse brain is a mouse brain.
So these organoids, these little mini brains, you're saying that they have all the types of neurons that you're going to see in the cortex of a human being. So they have the outer layers and the inner layers, and they have neurons, and they have all the astroglia. Do they have microglia?
Yeah, by all the sort of for the prediction or definition, they have a pretty good sort of layer structure. They all have a different type of neuron. And for sure, they have a neuron glia cell. And then they should have some microglia, but that's the area we have not studied enough. Right now we focus on just neuron and the glia. And then cortex is the most kind of studied portion of a brain organelle. But we are also working other portion of the brain organelle, like, okay, making the two brain region fuse together, like say, okay, fuse the cortical and the cerebellum, fuse the cortical and the hippocampus, fuse the cortical and the thalamic region. So that's the continued expansion. But for the cortex, it definitely is one much mature. You will see the structure, layer structure. You will see the cell type mimicking the human. Of course, it's the total cell number. It's less than the total brain cell for sure. But that overall structure mimicking pretty well for the human.
So how long can you keep them alive in the dish, these little brains?
Yeah, that's very curious, right? So as you know, human brain develop taking long time at the birth and go to the mature, take a year and a year. We cannot let that happen. So you just cannot wait that long. So in the petri dish, you definitely can move things fast. Typically, the maximum time we usually say, okay, the best time is roughly 90 to 120 days. That's the best time to study them. Before that, you kind of see sometimes, it depends on what experiment you do, right? Certain experiment you can do a little bit earlier when the organelle is in the 60 days, but a certain experiment you want to the organelle more mature, and we do later time. So the technical actually quite challenging. You can imagine we have to keep this one in the petri dish in the lab, not getting too contaminated, and keep they alive, kind of a good environment, so let them to be healthy and not cause any damage due to the condition we culture them. So technically, it's a labor-intensive work. It's also quite challenging. So, but we have a good set of people here at Yale, and we have a pioneer at Yale. So we believe we can do a very good job to making the system working the best. So, but it is a Terry, you're right. It's a good question. It's a technical challenge. As this is a new technique, as I said, it's only maybe 5 years, and we're getting better and better over the time. But it's not, definitely a lot of things can improve. It's not perfect, but.
So it's like taking care of a litter of puppies or something, right? I mean, you have to feed them and take care of them, change their water, the liquid that they're in, and all of that. It's a really it's care and feeding of these little, they look like little globs, right?
Like
they just look like,
we actually have very special material allowing them to live in, we call it gel type. Okay, just of course the people figure out before that. So they overall they are, as you have meticulous handle them, they are pretty good, pretty reasonable, survive very long. But it's a long experiment. You can see you start in the first day. You take 3 or 4 months to get into that shape. You can do something about it.
So do these little globs of brain cells that have all these layers, do they have seizures?
So they were mimicking the seizure activity. And for the Angelman, we have not studied them in the detail. So we had published one paper last year. So we definitely see some of this sort of seizure-like activity in this brain organelle. We studied one of them, okay, with my collaborator from Singapore. So we do see they may mimicking the brain have a we call hyper excitability. So then you can recording for. But then we definitely need more and looking at sort of more detail. It's last year we published it just the very beginning to using this organelle. This is the first time to show the potential you can use in brain organelle, actually testing one of a treatment and then see the suppression for this seizure-like activity in the petri dish. So.
Okay, so they have something that looks kind of like, you can tell that it's electrical activity that's kind of like a seizure.
Mm-hmm.
Do they have thoughts?
That's an excellent question. I don't have a good answer, direct answer for that. I would doubt for the sort for that they may have sort of we call a connectivity between the cell and the neuron. It's definitely talking each other, but that definitely among all the brain network together. But I doubt that will have mimicking the human sort of physiology, the thinking process. It's at least as far as I know right now, that's no one know what's the answer for now one, which is probably very, very unlikely. But they can mimicking a lot of brain activity, but not mimicking a lot of abstract. So the human psychological part of the process, like they will never. There's no language involved in this brain organ for sure.
So if you want to see if it's like Angelman syndrome, like the brains of people with Angelman syndrome, what you're looking for then are electrical changes or some kind of genes that go up or go down or some protein changes. That's how you're going to tell whether it's similar to a person or a mouse with Angelman syndrome, right?
Yeah, definitely you can do all molecular, all sort of expression, gene expression in a high resolution. Such as right now a lot of people have done this brain organelle, use the single cell level of an RNA expression analysis. So you can study very, very high resolution at the single cell level, see whether individual Angelman patients' neuron different from non-Angelman patients' neuron. So that's exciting experiment we propose to do. We want to do it. The second, it's you can definitely recording electric current from the cell by many different techniques. For example, you can just directly touch that single cell membrane to record it. Or the popular one right now we do, sort of using calcium as an imaging signal to recording them to sort of neuron activity in general, not just a membrane receptor current. And the third one is because we are capable right now making different layer or even different brain region fused together. So you can actually testing called the connectivity. See whether one brain region connect other brain region well or properly. So that's a pretty unique situation. You were never able to do this kind of thing before. All the time, you only can study single human neuron. For this brain organelle by name, because they are mini brain, so you can looking we call a connectivity, which is truly mimicking somewhere similar you do the human PET, human kind of life sort of function MRI, but then but you can manipulate. In the human brain, you cannot manipulate. You only can record. And here you can manipulate it. You record it first, then you put the treatment in. See whether that treatment is reversing the activity back to the normal or correct somewhere so the abnormal activity happened to the Angelman brain.
Okay, so if I summarize, one of the things you do is you can look at a single cell and see whether that single cell has different gene expression. And you can compare the Angelman single cell with a person who doesn't have Angelman, a human cell from somebody else. That's one way. Another way is you're actually looking at the connectivity from certain layers, or maybe, yeah, 2 different layers of the little mini brain. And what was the third way? It was the middle one that you said.
Yeah, middle one is you just recording the single cell for receptor.
Okay.
Yeah.
You want to see if the receptors change or move to a different location, or
yeah, the
if
current and the channel is open or closed or too excited, too open or too closed. So that's how you can do the single cell level traditional sort of electrophysiology. You just touch that receptor and channel recording them. That's the traditional technology. But this connectivity, you can look at 2 neurons, 2 synapses, how they talking to each other.
Very cool. And then you can throw medicine in the dish, yeah, with the mini brain, or maybe a genetic treatment, who knows what. Yeah. And you can throw that in the dish, or repurpose drug, or whatever it is that you're testing, and see if you can make the Angelman one look more like the one that doesn't have Angelman syndrome. Yeah, that's the beauty at the end. So you're asking me why we FAST want to invest into creating this biorepository. It's all the end to see if this reagent or biorepository you contribute eventually can lead and develop a new and a novel treatment or cure for the Angelman. Wow. Well, I wish I could sign up. My kid is over 22 already, but I hope that you are quickly able to collect all the cells that you need, because this is really going to help all of us. This is a fantastic thing that you're doing, and I'm really excited about it.
Yeah, that's my hope. We're definitely working hard, and I know you're all looking forward for whether we can something exciting and new. But I said this is the first step. Definitely multiple steps needed, but I'm definitely excited. This is a new tool and a new possibility and a possible new hope for the new treatment. So.
Well, I just want to tell you, it's been a pleasure knowing you for 21 years, and it's been
21. You're actually counting that. So yeah, well, definitely I'm surprised. I still remember I was talking to the other family. I say, oh wow, Terry Jo is a pretty unique, special person in the Angelman community for sure. And then when you and I first talking each other, looking for the mice, and you request the first mouse model from us, and I'm definitely, it's my sort of privilege kind of knowing you for many, many years too. So.
Wow. And what's really fantastic is that you're still working on the problem, and it's going to get fixed in your career, which is just absolutely amazing. So anyway, thanks for sticking with it.
Sure. Thank you for having me again. Good luck for everything.
Okay, and I hope we can talk again soon and hear an update about the mini brains. And anyway, thanks so much, and see you soon, I hope.
Bye-bye. Take care.
Bye.